This lecture introduces to the archaeobotany of humid tropical regions with a focus on the Americas and on Mesoamerica in particular. During the last decades, our knowledge on ancient environments and plant use in the humid tropics massively increased, stretching around the globe from Southeast Asia over the African tropical rainforest to Central and Northern South America. Climatically, these regions fall into the tropical and subtropical climate zones. Per definition and based on the Köppen climate classification, the tropical climate zones are characterized by temperatures above 18 degrees centigrade throughout all months of the year and are subdivided into three subtypes: the equatorial and tropical monsoonal regions, here in darker blue, and the adjacent tropical wet or dry savanna regions in lighter blue. Most significant here is the warm temperature, whereas humidity is variable with equatorial climates experiencing large quantities of precipitation all year round, and tropical wet and dry and tropical monsoon climates showing seasonal shifts in rain patterns. In focusing on the Americas, we find the tropical climate type mostly in northern Brazil, extended regions of Peru and Central America, including minor areas in the Gulf region of southern Mexico. The humid subtropical climate zone is characterized by hot and humid summers. These climates normally lie on the southeastern sides of the continents, adjacent to the tropical climate zone. Characteristic is at least 8 months with a mean temperature above 10 degrees centigrade. Monthly mean summer temperatures are normally between 24 and 27 degrees centigrade. Humidity, as classified by the Holdridge life zones, required that precipitation is always higher than potential evapotranspiration. Daily intense but brief convective thunder showers are common. These classifications coincide more or less with the Köppen definition of the tropical zones we saw in the last slide and the green shaded areas in this slide. In our focus on Central America, they include the central, more inland regions of Mexico adjacent to the coasts. The vast area of Mexico and adjacent parts of Mesoamerica are, apart from the tropical and subtropical climates, characterized by a tremendous range of other climate ranges, vegetational patterns, and topography, from volcanic mountain ranges to jungle-covered coastal plains. In agreement with the regional climate layout, palynological data indicate highly diverse environmental patterns in the different regions throughout the last 20,000 years. The reasons why we can consider archaeobotany of tropical regions to differ from the study of plant remains in temperate regions are manifold. First, the nature of plant resources for human and animal subsistence generally differs in accordance with the different climate zone and its predominant vegetation. In the tropics, the consumed biomass of food and crop plants derives to a major portion from the vegetative parts of the plants, whereas in temperate regions there's often a focus on seed plants, that is, cereals. They usually make a large proportion in the diet in regions where the vegetative biomass is reduced due to lower precipitation. The table to the right shows the major crops of tropical Colombia and France, with the latter of these dominantly falls into the temperate climate zone. We can easily recognize that in an archaeological context, most of the taxa in temperate regions would be preservable via their seeds. They are shaded in orange. In tropical regions, Most species would leave no seeds to be later found by archaeobotanists. Macro remains of roots, tubers, seedless fruits, and vegetables are generally underrepresented in archaeobotanical datasets because vegetative tissues, except wood charcoal, are relatively undifferentiated in their constituting plant cells, making identification of these parenchyma tissues extremely difficult. John Heather was among the first researchers addressing attention to the importance of recognizing vegetative food remains in archaeological sites and initiated the field of tropical archaeobotany with some important contributions in root and tuber identification. He was also author of a number of books in this field. Beside the very different geographic preconditions for the deposition of plant material, there are additional taphonomic factors that limit the preservation of vegetative plant remains. These are moisture in presence of oxygen, which acts decaying even on carbonized organic material. Therefore, the study of phytoliths plays a major role in sites of the humid tropics. Parenchyma tissue is not highly specialized and involved in many functions, such as photosynthesis, storage, synthesis and processing of many substances, and tissue repairing. As a simple permanent compound, it makes a major part of ground tissues in plants, where other compound structures like vascular tissues are embedded. They are non-vascular and composed of simple, living, and undifferentiated cells, which are modified to perform various functions. The other simple permanent compounds are collenchyma, such as the fibrous tissues in celery, and sclerenchyma, such as the stone cells in a pear. Further classification of parenchyma is based on functional characteristics, shape, and arrangement of the cells. Soft plant tissues generally undergo physical transformation on preservation, whether through charring, desiccation, or waterlogging. This is often in the form of expansion or shrinkage of the tissues, deterioration or loss of fragile parts, and fragmentation. This pattern of drastic changes contrasts markedly with that found in the archaeological remains of seeds and wood. Being relatively robust, the morphology of seeds and the anatomy and morphology of wood may change little on preservation, unless compressed. So that classical morphological and anatomical criteria may be used in their identification with only minor adjustments for the effects of charring. However, soft tissues are rarely preserved in a state that allows comparison with modern fresh material. For this reason, it is necessary to undertake the experimental charring of those plants that are represented in the archaeological record by highly deteriorated and fragmented remains. The effects of heat on the anatomy and morphology of fresh tissues may be compared with those expected in archaeological tissues. Not only classical characters, but also secondary characters resulting from charring may then be used in the description of similar archaeological tissues. The reconstruction of their original morphology and anatomy allows their identification by the comparison of diagnostic characters. The macroscopic characters of parenchyma tissues, such as cell shape and cell wall characters, provide important clues for the identification. However, basic research is limited, and unfortunately, not many researchers engage in this tedious type of identification of plant remains. Particularly, the archaeologically recognized scarcity of plant macro remains in humid or seasonally dry tropical and subtropical climates triggered the search for resistible plant microremains, such as phytoliths, pollen, and starch grains that can help to better understand ancient subsistence in these regions. One of the proponents of phytolith analysis in tropical environments is Dolores Piperno, who contributed a massive body of research on the paleoethnobotany of these geographical regions. She was also one of the first paleoethnobotanists who identified the earliest mice phytoliths in contexts from southern Central America and northern South America, dating from about 7,000 to 5,000 BP. She thereby proved that mice was domesticated and dispersed out of Mexico by the early 7th millennium BP into the seasonally dry tropical forest. This chronology also accords well with that derived from a molecular clock, which suggests that maize domestication occurred by around 8,000 BP, a basic research that has been conducted by geneticists at the University of Wisconsin-Madison, namely by John Dobley and his team. Primary basic research on mice phytoliths has been conducted by Deborah Pearsall, who was the first researcher to examine how mice phytoliths could be studied and identified in archaeological contexts. She compiled detailed size attributes of a type of grass phytolith called cross-shaped that is produced most commonly in leaves and also in lesser quantities in husks and tassels of the plant. In later research, Piperno investigated the morphological differences in cross-shaped forms from different grass species, including Teosinte, the wild progenitor of maize. Such studies involve, besides morphological differences, a considerable amount of three-dimensional measurements and statistical analysis of those. Before considering the emergence of early food production, we should have a look at the earlier time period, starting with the dispersal of Native Americans into Mesoamerica. Although the speed by which the Americas were populated, the number of basal divergences, and the degrees of isolation, mixture, and continuity in different regions are poorly understood, recent DNA studies provide a rough idea on the complexity of Native American dispersal and divergence throughout time. According to these results, ancestral Native Americans diverged from Siberians and East Asians around 23,000 years ago and split within that ancestral lineage between later Native Americans and ancient Beringians. After the initial entry, during which multiple splits of the population groups must have occurred, the southern Native American groups began to disperse across the North American continent between 14,000 to 6,000 years ago. Early South American populations possibly carried an Australasian-related admixture and diverged west and east of the Andes. After 8,700 years ago, movement out of Mesoamerica to the north and south took place, which was simultaneously accompanied by an inward gene flow from the north, visible in the genetic record only as unsampled populations. Alternative models suggest an arrival by boat. From time to time, there have been claims for a number of earlier settlements. Most recently, human occupation at around 33,000 years ago in Chiquihuete Cave in northwestern Mexico has been stated, which would considerably predate the data received from modern population by molecular dating. Conservatively, the first people in Mexico are placed at 11,500 years ago, since Clovis points of the early Paleo-Indian period have been found scattered across Mexico, Guatemala, and Belize, and into Central America. Later Paleo-Indian cultural remains are generally harder to recognize, and sometimes have very limited distribution in Mesoamerica. Consideration must be given to pre-Clovis sites, sometimes called the pre-projectile point stage, prior to 11,500 BP. The finds at Monteverde in Chile have been widely accepted by New World archaeologists and now push Paleo-Indian chronology back to about 18,000 to 20,000 years ago. Claims have been made for sites of this age or of even greater antiquity in Mesoamerica, but most fail to meet basic geological and archaeological standards of credibility. One early argument for great antiquity of human cultures in Mesoamerica resulted from a discovery in 1870 of the sacrum of a Pleistocene camelid found at Tequixquiac, northwest of Mexico City. The sacrum had been slightly carved to represent an animal. To my knowledge, the specimen has never been directly dated, but is thought of having a maximum age of 14,000 years. Loltun Cave in Yucatan is perhaps the best candidate for a Paleo-Indian age. There is a variety of late Pleistocene fauna, including mastodon and horse, along with modified stone and bone objects. While the faunal stratigraphy goes back to an age as far as 30,000 years, human occupation probably took place not before 14,000 BP. Early Holocene, that is, late Paleo-Indian cultural patterns dating between 8,200 and 6,800 BC, that are common in the southern plains and throughout North America, are very difficult to recognize in Mesoamerica. Lerma projectile points, as discovered in Tamaulipas as well as in the Tehuacan Valley, belong to the transitional period from the Paleo-Indian to the Archaic period. With the end of the Pleistocene, the megafauna starts disappearing, which had often been related to humans overkilling these large animals, but alternatively also to climate change. Recent research using some calibrated radiocarbon date distributions to reconstruct population levels of megafauna and humans suggests that the causes for extinctions varied across taxa and by region. In 3 cases, extinctions appear linked to hunting, while in 5 others, they are consistent with the ecological effects of climate change, and in a final case, both hunting and climate change appear to have been responsible. The picture to the right shows the results of these studies with the megafaunal population busts in red by region and in relation to the Clovis period and the Younger Dryas. In focusing on the time of rising cultivation and transition to domestication, we consider here the late Paleo-Indian, the early Archaic, and the Middle Archaic periods, and leave out the later rich and widely known cultures of the formative and classical periods. Although the beginnings of agriculture are generally set to the early Archaic period, the patterns of the transition from predominantly hunting and gathering to agriculture are still patchy. The problems hindering the development of a coherent picture of change can be summarized to lay in the small number of known sites, the disappearance of lowland coastal sites through the rising sea level at the end of the Pleistocene, the lack of suitable deposits either in depth or extension, and the lack of archaeobotanically investigated deposits. As a general climate trend at the end of the Pleistocene, we realize a distinctive reverse development in moisture along the 20th degree of northern latitude, which separates a moist late glacial and dry Holocene climate north of it from a dry late glacial and moist Holocene south of it. This indicates increasingly improving conditions for plant growth in Mesoamerica with the end of the Pleistocene. These major ecological changes are particularly prominent when considering the consequential developments in human subsistence behavior that comprise hunting techniques and tools adapted to smaller animals, smaller and broader projectile points, grinding stones, and first preserved baskets for cooking and storage. The Archaic period is usually subdivided into three subperiods: the early, the middle, and the late Archaic. The early Archaic, from about 8000 BC, provides the first evidence of cultivated species of the cucurbit family and of maize. During the Middle Archaic, agriculture becomes established and further expands during the Late Archaic period. The Early Archaic is represented by 4 or 5 traditions from north to south. The Cochise tradition of the southwestern United States and far northwestern Mexico, the Infanillo and Big Bend traditions on the northern periphery of Mesoamerica, the Santa Marta and El Riego traditions, and the southern maritime tradition. The El Riego tradition, from about 7600 to 5000 BC, had a seasonally scheduled subsistence system with hunter-gatherer tools and milling stones. The closely related Santa Marta tradition, from about 8000 to 4000 BC, extends from Santa Marta Cave to Oaxaca, and Cucurbita pepo and siamais were either collected from the wild or already in the first stages of domestication in Oaxaca. Tamaulipas sites that are ascribed to the Infanillo tradition had abundant vegetal artifacts such as nets, mats, baskets, and plant remains that included pumpkins, so in this case Cucurbita pepo, gourds like Lagenaria siceraria, and possibly chili peppers. We already heard in our lecture on African archaeobotany about the difference of the term domestication, which refers to genetic changes in a population as a result of human selection, either conscious or unconscious, and the term cultivation, as the act of encouraging the growth of certain plants through improving the soil, eliminating competing plants, transplanting, watering, and fertilizing. Although this distinction appears to be straightforward, human subsistence systems lie at points along a continuum between foraging and agriculture. Domestication and the long process of the development of an agricultural way of life represent a long history of interaction between people and plants, today often referred to as coevolution. This process is also recognizable in highland and lowland Mesoamerica. Climatic change at the transition from the Pleistocene to the Holocene dramatically altered plant communities and also brought about a rise of the sea level that created new ecosystems along the Mesoamerican coast. Although these environmental changes may not provide a satisfactory explanation for plant domestication and the development of agriculture, they did present new challenges and opportunities to the early occupants of Mesoamerica. A systems theory approach has been adopted by Kent Flannery to understand the process of plant domestication and the development of agriculture in the highlands of Mexico. According to Flannery's model, hunter-gatherers gradually adapted to changes in plant communities during the Pleistocene to Holocene transition, making increasingly use of wild species, which they eventually domesticated. Investigation of ancient seasonal encampments and settlements and the recovery of associated plant and animal remains have allowed the reconstruction of subsistence changes in the highland valleys of Oaxaca and Tehuacan. The onset of the Holocene included a general shift from large game hunting to smaller game and the increasing exploitation of plant resources, also referred to as the broad-spectrum revolution. Considering centers of domestication, it is interesting to note that the equatorial humid tropics are generally not indicated as primary centers where crops were taken into domestication, but rather as regions into which agriculture spread from adjacent centers. The basic map of the one you see here goes back to Harlan and Vavilov, but has been further developed by Fuller and colleagues. Accepted primary domestication centers are shown in black, and potentially important secondary domestication centers are pictured in gray. The hatched areas show quite recent research results, mostly based on archaeobotany. Arrows indicate major trajectories of spread of agriculture and crops out of some centers. For Mesoamerica, we can note the domestication of Cucurbita pepo as well as siamase going back as far as 10,000 years BP. From there they dispersed into other regions such as the northern lowland neotropics. Questions relating to the origins and early history of maize have seen decades of research by investigators representing diverse scientific disciplines. However, adequate chronologies for the earliest utilization and cultivation of teosinte and its transformation under human selection into maize are still to be determined by archaeological research. Two factors that bear significantly on these issues have been extensively researched during the past 20 years, providing indications that archaeobotanical data on the earliest history of maize evolution in Mexico may become available before too long. One was that the cradle of maize domestication, long thought to be the arid Mexican highlands, is considered now, on the basis of molecular studies of modern wild and domesticated varieties of Sea, to be located in the more humid, lower elevation central Balsas River Valley of tropical southwestern Mexico. These results have been produced by the team of John Dobley at the University of Madison, Wisconsin. There are a number of instructive YouTube videos on their research of particular interest to those of you with special interest in the genetics of crop plants. The molecular results conform with existing archaeological data from Mexico. The earliest maize cobs recovered from the dry highlands outside the present-day range of wild maize date to 5,300 BP. They are already completely domesticated. No evidence for an earlier utilization of teosinte, the wild progenitor of maize, exists, as would be expected if maize were of highlands origin. Therefore, 5,300 BP is the terminus antiquem teosinte must have been brought under domestication, while this process did probably not start before 10,000 BP. Teosinte is the common name of a group of 4 annual and perennial species of the genus Sea native to Mexico and Central America. Teosinte plants are taller and broader leaved than most grasses. Their general growth form is similar to that of maize, although they have much longer lateral branches. The name teosinte is of Nahuatl Indian origin, and it has been interpreted to mean grain of the gods. Some species of teosinte are distinct from maize, both genetically and taxonomically, and they appear not to have played any role in the origin of maize. However, one form of teosinte, known as siamase subspecies parviglumis, shares a particular close genetic relationship with maize, and available evidence indicates that it is the direct ancestor of maize. The parviglumis subspecies grows in the valleys of southwestern Mexico. In these regions, it grows commonly as a wild plant along streams and on hillsides, although it can also invade cultivated fields as a weed. It is most common in the Balsas River drainage of southwest Mexico and hence is also known as Balsas teosinte. While the maize and teosinte plants share a similar robust growth form, their female inflorescences or ears are strikingly different. The teosinte ear possesses only about 5 to 12 kernels, each sealed tightly in a stony casing. Collectively, the kernel and its stony casing are known as fruit case. At maturity, the teosinte ear disarticulates such that the individual fruit cases become the dispersal units. Protected within its casing, the teosinte kernel can survive the digestive tracts of birds and grazing mammals, enabling the seed to be easily dispersed. By comparison, the massive maize ear can bear 500 or more kernels, each of which is attached to the central axis of the ear or cob. The kernels are naked without adequate protection from predation and are easily digested by any animal that consumes them. Since the kernels are firmly attached to the cob and the ear does not disarticulate, a maize ear left on the plant will eventually fall to the ground with its full suit of kernels. When hundreds of maize kernels germinate the next season, so close to one another, the emerging plants are unable to obtain adequately light and soil to grow and produce. Thus, maize is completely dependent on humans for its survival, as all grain crops are. The creation of maize was a more complicated process than the domestication of other cereals, because its derivation from teosinte involved changes under human selection pressure to 5 major regions of the Sia genome that governed the plant's architecture, cob structure, and row and kernel number. These phenotypic alterations constituted the greatest known between any wild progenitor and its domesticated product in all of prehistoric agriculture, as we will see in a later lecture on Western Asian domestication of wheat and barley. It is this fact of the massive phenotypic alteration accompanying maize domestication that forms the basis for successful comparative studies on maize and wild grass phytoliths. Some scholars question whether teosinte could be a useful grain crop because its seeds are tightly encased in a hard, woody cupulate fruit case, suggesting that the ancient peoples would have been incapable of freeing the grain from this casing. Their alternative explanation was that early wild maize usage was focused on the consumption of the sweet, immature ears of the plant as vegetables or for turning the sugar of the stalk into fermentable beverages. This idea was eagerly taken up by some few scholars who argued on a more general level that the wild progenitors of cereals were originally used to produce alcoholic beverages to be used in rituals. Such models are often related to social anthropology and the idea of a probably overestimated role of feasting in ancient societies. However, if such use of the grains would have been widespread in the past, then teosinte fruit cases, together with their phytoliths, should be largely absent from early Archaic period sites in the Balsas River Valley, whereas the practice of using the ripe grain as food should be detectable in the presence of phytoliths from the fruit cases of teosinte on stone tools. Despite the massive progress in phytolith and starch research during the last years, this question has not been solved yet conclusively. Dobley presents convincing arguments for the use of the grains for food purposes, which are provided by the fact that the earliest archaeological corn, which is some 6,000 years old, has grains that are free of the fruit case, indicating that selection for edible grain happened very early. He also did experimental harvests on teosinte to calculate the yield of it. He recovered an average of more than 1,000 fruit cases per plant, which equals about 36 grams of fruit cases. Since about half of the weight of the fruit case is composed of the grain, the average plant yielded about 18 grams of grain or flour. The yield overall of Doblay's plot was more than 400 kg per hectare, a value that is not too far off yields of 1,000 kg per hectare of early US open pollinated maize varieties. In conclusion, Dobley, Flannery, and other researchers come independently to the result that teosinte is a high-yielding plant with the potential for a small plot to support an ancient community. The spread of maize from its domestication center in the Balsas Valley into South America could be traced by DNA analysis on historic maize specimen, indicating an expansion route across the highlands. Deviations in the timing of the arrivals in different regions may also be explained by multiple connection routes. Squash seeds, peduncles, and fruit rind fragments from archaic stratigraphic zones of Guilanaquith in Oaxaca are assigned to Cucurbita pepo on the basis of diagnostic morphological characters and identified as representing a domesticated plant on the basis of increased seed length and peduncle diameter, as well as changes in fruit shape and color in comparison to wild Cucurbita gourds. 9 accelerator mass spectrometer radiocarbon dates on these specimens document the cultivation of Cucurbita pepo by the inhabitants of Guilanaquith Cave between 10,000 to 8,000 calendar years ago. Other sites of the regions, Coxcatlan and San Marco Caves in Puebla, and Romero's and Venezuela's caves in Tamaulipas, provided similar old dates for the domestication of this species. Cucurbita pepo comes with an extensive range of cultivars today, including pumpkin, zucchinis, marrows, as well as many types of squashes and ornamental gourds. It is important to note that the common names pumpkin and squash are often also used for other Cucurbita species, such as the fruits of cultivated forms of Cucurbita maxima and Cucurbita muscata. Cucurbita pepo is a species of great economic importance, and mature and immature fruits, flowers, and young leaves are used as vegetables. The large seeds are also edible and rich in zinc, and there is a long history of medicinal use in Central and North America, where sap and pulp are applied to burns and seeds are used as a diuretic. As in the case of wild and domesticated maize, phytoliths from the New World genus Cucurbita appear to be highly useful in documenting the earliest history of domesticated squashes and gourds in the Americas. All parts of the Cucurbita plant make high amounts of phytoliths, but the most diagnostic and useful ones originate in the fruit rind. Bossarth first observed and described these large spherical phytoliths and noted how they had scalloped surfaces of deeply contiguous concavities, now called scalloped phytoliths. Large surveys of wild and domesticated Cucurbita and other tropical Cucurbitaceae, carried out by Piperno, confirmed the genus-specific taxonomy of Cucurbita scalloped phytoliths and indicate that those from wild Cucurbita are frequently much smaller than those from domesticated varieties, suggesting that certain species of wild and domesticated Cucurbita are identifiable on a morphological basis. It is meanwhile also known that in at least 2 major American crop plants, Sia and Cucurbita, phytolith formation is under the control of genetic loci that accounted for significant phenotypic changes in the fruits and in their phytoliths during the evolution of the domesticated species from their wild progenitors. Other genera of the tribe that Cucurbita belongs to, the Cucurbitae, such as Lagenaria, also contribute types of scalloped phytoliths, but these are distinctive from those of Cucurbita, as you can see in the left picture, in B. There are, as the map clearly shows, more centers of domestication in the Americas, such as the northern lowland near tropics, visible here in 2A. Here, Cucurbita muscata, which is another pumpkin species, Ipomaeo batatas, the sweet potato, and Phaseolus vulgaris, the common bean, were domesticated at around 9,000 to 8,000 BP. Beyond these regions, the central mid-altitude Andes, the north and central Andes, lowland southern Amazonia, Ecuador, and northwest Peru represent additional centers of domestication. The almost endless number of species that has been domesticated in the Americas, mainly in the later periods, are left without comment to you in the last 3 slides.